Polio vaccine incident. In 1955, polio was considered one of the most terrifying diseases of that time, especially in the United States. This viral disease mainly targeted children and attacked the nervous system. The virus eventually reached the spinal cord and caused sudden paralysis. In the most severe cases, the respiratory muscles would fail, and the patient would need devices such as an iron lung to survive. During the 1940s and 1950s, large waves of this disease appeared every summer across the United States and Europe. Even the President of the United States, Franklin D. Roosevelt, was one of the most well-known victims of this disease. During this period, a scientist named Jonas Salk worked for about 7 years on developing a polio vaccine. His method was to take the polio virus and inactivate it using chemicals so that it could no longer cause disease, but would still be able to stimulate the immune system and make the body resistant to the virus. Although this idea seemed simple in theory, its implementation was extremely sensitive and complex, because if even a small amount of active virus remained, the vaccine itself could turn into a cause of the disease. In 1954, one of the largest medical trials in history was conducted, and more than 1 million children participated in it. After the vaccine was announced to be effective in April 1955, it was decided to produce it on a large scale so it could be distributed to millions of children. For this purpose, several pharmaceutical companies were responsible for its production, one of which was Qatar Laboratories. It was at this stage that one of the biggest mistakes in medical history occurred. The virus inactivation process at this company was not properly carried out, and in some vaccine vials, the virus remained alive and active, but this defect was not detected in quality control tests. In April 1955, about 200,000 children in several U.S. states received this vaccine. It was later discovered that this faulty vaccine caused around 40,000 cases of polio infection. Among these cases, more than 200 children developed permanent paralysis, and about 10 people lost their lives. Even worse, the virus was transmitted from these children to others, and the disease spread further. This was because the polio virus replicated in the intestines and was excreted through the stool of an infected person. It could then enter another person's body through contaminated hands, water, or food. The elixir sulfanilamide. Diethylene glycol, DEG, is a toxic yet sweet-tasting chemical substance that is used in products such as antifreeze, car brake fluids, industrial solvents, and some other chemical materials. In 1937, the pharmaceutical company S.E. Massengill used diethylene glycol as a solvent or excipient and produced an oral sulfanilamide drug, which they named Elixir sulfanilamide. DEG was a toxic chemical substance to humans and other mammals, but Harold Watkins, the company's chief pharmacist and chemist, was not aware of its toxicity and potential dangers. Although the first death linked to ethylene glycol was reported in 1930, and some medical studies had already indicated that DEG could cause kidney damage or failure, its toxicity was still not widely recognized at the time. Watkins added raspberry flavor to improve the taste of the drug, and then dissolved the mixture in DEG. At that time, animal testing was not required, and Massengill did not conduct any safety tests, as there was still no law requiring companies to test drug safety before releasing products to the market. The sale and distribution of the drug began in September 1937. By October 11th, the American Medical Association had reported several deaths caused by the drug. After that, the U.S. Food and Drug Administration took action. Investigations in a research project showed that DEG was the main cause of the fatal side effects, and an extensive effort was made to recall the distributed drug. In the end, at least 100 deaths were attributed to this medication. The owner of Massengill, under pressure, denied responsibility and stated, "We only carried out a professional request and could not have predicted the results." It was later revealed that the company's safety tests had only examined appearance, taste, and smell, not actual toxicity. In the end, Watkins, the chemist responsible for the case, ended his life before the trial. The Thalidomide tragedy. Thalidomide was first developed in 1953 by a Swiss pharmaceutical company as a sedative drug. In 1954, the company stopped its development, but later the drug was acquired by a West German pharmaceutical company, which continued the development process. In 1957, thalidomide entered the market and was introduced as a medication for treating anxiety and insomnia. It was also prescribed to help reduce nausea during pregnancy, even though it had not been adequately tested on pregnant women. At first, the drug was believed to be safe during pregnancy, but in 1961 concerns about its connection to birth defects began to increase, and it was removed from the European market in the same year. Thalidomide was used in 46 countries, and its consequences became one of the biggest drug-related tragedies of the 20th century. When used during pregnancy, the drug was associated with abnormalities, such as incomplete or abnormal development of the limbs, bone problems, and changes in the formation of the hands and fingers. In some cases, problems affecting areas such as the face, ears, eyes, and internal organs including the heart and kidneys were also reported. Estimates show that more than 10,000 and possibly up to 20,000 fetuses were affected by thalidomide exposure during pregnancy. Among them, around 40% of affected babies died at birth or shortly afterward. Research showed that the timing of thalidomide use during pregnancy played an important role in the type of abnormalities that occurred. Taking the drug around the 20th day of pregnancy could affect the development of the nervous system, while days 21 and 22 could affect the development of the eyes, ears, and face. In later stages, around days 24 to 28, the development of limbs such as the arms and legs could also be affected. This tragedy led many countries to establish stricter rules for drug testing and the approval of new medications. Tuskegee syphilis study. At that time, a disease called syphilis was widespread among people. It was a contagious, infectious disease that, if left untreated, could gradually affect different parts of the body, including the skin, brain, and heart, and could eventually lead to death. In this context, a study was conducted between 1932 and 1972 by U.S. government health organizations. In this study, 600 African American men were enrolled. 399 of them had syphilis, while 201 were healthy. To encourage their participation, they were promised free treatment, but they were not informed about the true purpose of the study. Despite risks such as blindness, deafness, heart disease, mental disorders, and death, the condition was kept hidden from the participants. The real goal was to observe the natural progression of the disease without treatment, including until death and autopsy. At first, the participants were told that the study would last only 6 months, but in reality, it continued for 40 years. During the early months, the participants were examined and then received some limited treatments, which in some cases were toxic. However, later they were given placebos, and some diagnostic procedures, such as spinal fluid sampling, were introduced to the participants as treatments. Although penicillin had been available for treating syphilis since 1945, this treatment was intentionally withheld from them in order to study the natural progression of the disease. In 1972, a whistleblower named Peter Buxton brought this case to the media, and the study was eventually stopped. By the end of the study, only 74 participants were still alive. 28 people died directly from syphilis, and around 100 others died due to complications caused by the disease, including heart damage, neurological and brain problems, and severe infections. Additionally, 40 wives of the participants became infected with syphilis, and 19 children were born with congenital syphilis. Thanks for watching until the end. Make sure to like and subscribe for more videos.